July 28, 1976. Cries Planitia, Mars, 8 days after Viking 1 became the first American spacecraft to land successfully on the Red Planet. A mechanical arm delivered a small sample of Martian soil into a sealed laboratory. The machine added 1 drop of liquid. Inside that drop were simple nutrients, each tagged with radioactive carbon. If something in the soil consumed them and released gas, Viking would hear the radioactive signal. Within hours, the detector began to climb. Not a random spike, a strong response that continued for days. When the experiment was repeated at a second landing site, thousands of kilometers away, the line rose again.
Then came the control. Viking heated a duplicate sample to a temperature designed to destroy microorganisms. The strong response disappeared.
Feed the soil, and gas appeared. Heat the soil first, and it did not. That was exactly the kind of contrast the experiment had been built to recognize.
But Viking did not announce life on Mars. The mission reported unusual chemistry, not living organisms. And 50 years later, NASA has never sent another mission to Mars that directly tests for life existing there now.
So what did Viking actually detect? A Martian metabolism? Or a chemistry so unfamiliar that it could imitate life inside the first laboratory ever operated on another world? In 2026, NASA marked 50 years since Viking reached Mars. The anniversary celebrated a mission that transformed planetary exploration. It also returned attention to an uncomfortable sentence on NASA's own life detection website.
Direct detection of extant life has not been attempted by NASA since the Viking missions of the late 1970s.
Every successful rover since Viking has looked for a different part of the story. Ancient water, habitable environments, organic molecules, or possible signs that life existed billions of years ago. None has repeated Viking's attempt to ask whether Martian soil is alive today. That does not mean NASA abandoned the question out of fear. Viking taught scientists that before a machine can identify alien biology, it must understand every ordinary chemical process capable of producing the same signal.
And in 1976, the chemistry of Martian soil was almost completely unknown.
Viking 1 touched down on July 20, 1976, at 7:53 in the morning Eastern Time. The confirmation took another 19 minutes to cross more than 200 million miles of space.
Touchdown, we have touchdown.
We didn't know.
25 seconds after landing, the spacecraft began taking its first picture. The image did not arrive as a photograph. It assembled itself line by line on monitors at the Jet Propulsion Laboratory until a rock appeared, then the footpad, then the first landscape ever seen from the surface of Mars. The lander carried cameras, weather sensors, a chemistry instrument, and a robotic scoop. But the most ambitious hardware sat behind 4 small openings on its upper deck, a miniature biological laboratory sterilized more aggressively than any American spacecraft before it.
The scientists had 2 problems. They could not contaminate Mars with Earth microbes, and they could not allow an Earth microbe hiding on Viking to become the first false Martian.
The assembled lander was baked at roughly 111 degrees Celsius for 40 hours. Then, on Mars, its arms scraped the surface and deposited soil into instruments designed around different ideas of what alien life might do. The gas exchange experiment gave Martian soil water and nutrients, then measured the gases above it. A living community might consume one gas or produce another. The pyrolytic release experiment exposed soil to light and an atmosphere containing radioactive carbon. If something behaved like a photosynthetic organism, some of that carbon might be incorporated into new material. The labeled release experiment reversed the exchange. It placed radioactive carbon inside a nutrient solution and asked whether the soil would release radioactive gas after being fed.
A 4th instrument, the gas chromatograph mass spectrometer, was not a life detector. It searched the soil for organic compounds, the carbon chemistry that living systems require and that meteorites should deliver even to a lifeless planet.
Together, the instruments were supposed to form a chain, detect metabolism, confirm that the response changed when the sample was sterilized, then find the organic material that could support the biological interpretation. Only one part of that chain would behave as expected. In the labeled release chamber, the active Viking 1 samples climbed rapidly to thousands of radioactive counts per minute. The response slowed, but it did not vanish. A fresh sample tested later produced the same basic pattern. Viking 2 landed at Utopia Planitia on September 3. Its soil produced a remarkably similar evolution of radioactive gas. One active sample was collected from beneath a rock, away from direct ultraviolet light, and it responded too.
That repetition matters. A damaged instrument can create a strange reading. Two instruments on two landers reacting to different samples are harder to dismiss as a simple malfunction.
Gilbert Levin, who designed the labeled release experiment, and Patricia Strott, his co-investigator, compared the curves with terrestrial soils and even lunar material. Their 1976 paper described an active agent stable at cool temperatures, weakened by 3 hours at 50 degrees Celsius, and completely inactivated at 160. They did not claim the case was closed. Their paper stated that the available facts did not yet permit a conclusion about life on Mars.
But the result had crossed an important line. It no longer looked like a dead sample doing nothing. Something in Martian soil was reacting quickly and repeatedly to the experiment.
A control experiment is supposed to remove the factor being tested while leaving everything else as similar as possible. For Viking, heat was the key. On Earth, heating soil to 160 degrees Celsius destroys microorganisms and biological activity. Viking heated a portion of Martian soil, allowed it to cool, added the same nutrients, and monitored the same detector. The contrast was dramatic. The active samples rose toward 10 or 14,000 counts per minute. The fully heated Viking 1 control remained near the bottom of the graph.
That is the strongest part of the case for biology. The response was not only activated by nutrients, it was removed by a treatment expected to remove life.
But heat is not exclusive to biology. It can decompose unstable chemicals, change mineral surfaces, and disable an oxidant. A heat-sensitive reaction can imitate a heat-sensitive organism.
The control told scientists that the active agent could be destroyed. It could not tell them whether that agent had ever been alive.
The decisive objection came from the gas chromatograph mass spectrometer. Viking heated Martian soil in small ovens and analyzed the gases for organic molecules. It found water, carbon dioxide, and traces of chlorinated compounds that were initially treated as contamination. It did not find the broad inventory of organic material many scientists expected in soil containing microorganisms.
If life was consuming the nutrients, where was the carbon that life was made from? And if meteorites constantly deliver organic material to Mars, why did the soil appear chemically empty?
The other biology experiments added more uncertainty. Gas exchange released a surprising burst of oxygen, but its later behavior looked more like reactive soil than sustained metabolism. Pyrolytic release produced a small signal, yet the result was difficult to reproduce and interpret. The full package did not tell one coherent biological story. One experiment resembled metabolism. The chemistry instrument found no convincing organic foundation beneath it.
Most of the scientific team therefore judged a non-biological reaction more likely, although the mechanism was not proven.
Mars has almost no protection from ultraviolet radiation. For millions of years, sunlight and cosmic radiation have attacked the upper surface, creating reactive compounds in a cold, dry environment. When Viking added liquid water and organic nutrients, it may have triggered chemistry that almost never occurs naturally on the present surface. The experiment could have manufactured the very reaction it interpreted as metabolism.
Then there was the second injection. In a simple biological model, adding more food while organisms remain active should produce another increase in gas.
The later Viking sequence did not show a clean second surge, but the sample had been stored for months. Its active response had already weakened, and the flight report recorded uncertainty about nutrient delivery. The result challenges biology without providing a perfect chemical answer. Scientists proposed hydrogen peroxide, superoxides, iron oxides, and other reactive materials. Each explanation reproduced parts of the behavior. None turned the entire Viking result into a simple, universally accepted laboratory trick.
The strongest chemical suspect would not be discovered on Mars until 32 years later.
In 2008, NASA's Phoenix lander found perchlorate salts in Martian soil. Perchlorates can remain stable in the cold, but become highly reactive when heated. That discovery changed the interpretation of Viking's missing organics. The mass spectrometer had searched for carbon compounds by baking the sample. If perchlorate was present, the oven could release oxygen that destroyed much of the organic material before the instrument detected it. Laboratory experiments with Mars-like soil showed that heating organics with perchlorate can convert them into carbon dioxide and small amounts of chlorinated compounds, including the same kinds Viking had dismissed as terrestrial contamination.
This creates the Viking paradox. The instrument that appeared to prove the soil contained no organics may have erased them during the act of searching.
Perchlorate also supports a reactive chemistry interpretation of the biology experiments. It weakens the old argument that Mars was chemically empty, while strengthening the argument that Martian soil can behave in ways Vikings designers did not anticipate.
The later discovery did not settle the case. It made both sides more complicated.
Since Viking, missions have found overwhelming evidence that ancient Mars had rivers, lakes, groundwater, and environments that could preserve carbon chemistry for billions of years. Curiosity detected organic molecules in mudstone inside Gale Crater. In April 2026, after years of analysis, NASA reported the most diverse collection yet found in a single Martian sample: 21 carbon-containing molecules, 7 detected on Mars for the first time.
One was a nitrogen-containing ring related to structures that can precede more complex biological molecules. But organic does not mean alive. Geological reactions and meteorites can also create and deliver carbon compounds.
The discovery does not prove that Viking found microbes. It does prove that the absence of organics in the 1976 ovens was not the final verdict on Martian carbon chemistry. Viking sampled only the exposed surface at 2 locations. Modern missions increasingly search in ancient rocks, beneath weathered surfaces, and in places selected because water once concentrated and protected potential evidence. The biological case begins with the data itself. Strong gas evolution appeared at 2 distant landing sites. Fresh samples responded repeatedly. Material collected beneath a rock responded. Heating weakened or eliminated the effect.
And later missions removed one of the original objections by confirming that organic molecules can survive on Mars and that Viking's heating method may have hidden them.
The chemical case begins with what Viking did not find. No organism was seen. No uniquely biological molecule was detected. The other experiments did not corroborate a living system, and the surface is filled with reactive compounds unknown when the instrument was designed.
A positive-looking metabolic signal is not enough when non-living chemistry can use the same pathway to the detector.
This is why the word positive can mislead. The instrument recorded a positive response to its nutrient. It did not identify the agent producing that response. Gilbert Levin spent the rest of his life arguing that the biological interpretation best fit the complete pattern. Many Mars scientists concluded that chemistry was more likely. Both positions begin with the same curves. They disagree about what kind of world could have produced them. Viking did not prove that Mars is alive. It demonstrated something more frustrating. A machine can meet several expectations for life and still fail to distinguish life from an unfamiliar planet.
That failure changed the strategy of Mars exploration. NASA stepped back from direct detection and first asked whether Mars had ever been habitable, whether water existed, whether organics survived, and where evidence would be best preserved.
That slower strategy has transformed the planet from the sterile desert imagined after Viking into a world that once held lakes, complex chemistry, and environments potentially suitable for microorganisms. But the original question remains open. Not whether ancient Mars could support life, whether anything is living there now, below the radiation-damaged surface in ice, salt, or protected pores inside rock.
NASA's modern framework explains how future missions should demand multiple independent signs: metabolism, structure, chemistry, context, and controls capable of excluding every known non-biological process.
50 years ago, Viking carried one of the boldest experiments ever launched. It fed Martian soil, watched a line rise, and discovered that finding a signal is easier than proving what made it. The most extraordinary part of the story is not that Viking answered the question. It is that half a century later, we still have not asked the same question with better tools.